Hello everyone, this is video number 3 for the urinary system. In this video, I'm going to describe in detail some of the microscopic anatomy inside the kidney. And this has to do with the main functional unit in the kidney, the nephron. So last time we talked about part of that structure, and that's called the renal corpuscle. That contains the blood capillaries that are going to be filtered, producing a substance called the filtrate. Now, the filtrate enters the other part of the nephron, which is the main topic for today, and that is the renal tubule. So, just to look at the outline here, this number 2 on the outline is kind of a mixture of the two topics that I mentioned, the glomerulus and these blood vessels we talked about last time. And the tubule is the main topic for today. All right, so just looking at this slide, which was from the previous video, video number 2. Here are the blood vessels bringing blood in, afferent arteriole. Here are the glomerular capillaries. Glomerulus is the name for the nest of capillaries here. That's surrounded by a glomerular capsule, which has an outer layer and an inner layer. The outer layer is the shell, and the inner layer is stuck on the glomerular capillaries. Material is going to filter out of the blood into this capsular space, and then it's going to start moving down this tubule. So this is the renal tubule. It has 3 main segments. The first, closest to the glomerulus, or the corpuscle, is the proximal convoluted tubule. Proximal because it's close, convoluted because it's very twisty-turny. Okay? So all of these structures are in the cortex of the kidney. This tubule is going to connect to the next segment, the loop of Henle. This is basically one long loop that descends down into the medulla, and then another loop that ascends out of the medulla. So that's the loop of Henle. That leads to the third segment, which is a distal convoluted tubule. This is very twisty and turny, just like the other segment, but it's far from the corpuscle, distal convoluted tubule. That's going to lead into a very small segment called the collecting tubule, and then to a large vessel called the collecting duct. Okay? So we're going to go through these different segments and talk about the structure a little bit, and then talk about the main idea about what's going on in those segments. And then that'll lead us into a discussion about the physiology of urine formation. All right, so the renal tubule altogether is, go back. The renal tubule altogether is made up of different segments, the PCT, the loop of Henle, the DCT, through which filtrate from the blood is going to pass. The entire wall of this tubule is made up of a single layer of epithelial cells. And these cells are mostly cuboidal. If you look in the PCT, single layer of cuboidal cells. If you look in the DCT, also single layer of cuboidal cells. The loop of Henle, however, has 2 main segments, a descending loop, descending into the medulla, and an ascending segment, which is coming back out of the medulla. And within this part of the loop, there is a segment that's relatively thin compared to the other parts of the tubule. So that's called the thin segment. The thin segment, unlike all the others, is lined by a simple squamous epithelium. And then the thick segment is going to be similar to these two. And the collecting duct, also similar to proximal and distal convoluted tubules, in that it's lined by a simple cuboidal epithelium. All right, so let's talk about the 3 main segments of the renal tubule, starting with the proximal convoluted tubule, and talk a little bit about their structure and the main function that's going on there. The proximal convoluted tubule the wall of that tubule is made up of a single layer of cuboidal epithelial cells. And if you look at those cells, they have a number of microvilli on the luminal surface of the tubule. So these microvilli are facing the lumen, and whenever you see a cell with microvilli, you should automatically know that this cell is involved in absorption. Microvilli increase the surface area of the cell and make it very good at absorption. And that's what the cells lining the wall of the PCT do. The filtrate that passes out of the blood into the capsular space in the Bowman's capsule is going to go into the PCT. And a number of things from that filtrate are going to be returned to the body by these cells in the wall of the PCT, absorbing them and transporting them out of the tubule. Okay? So that makes the cells of the wall of the PCT very, I guess you could say, efficient at absorbing materials in the filtrate that are meant to be returned back to the body instead of passing into the urine. Okay? Next segment, the loop of Henle. So I think I've described this mostly already. Two portions, a descending segment and an ascending segment. The descending segment has a different permeability than to the ascending segment. The descending segment is permeable to water, but not to sodium or chloride ions, whereas the ascending segment is just the opposite. And I did mention already that the thick parts of the loop of Henle are lined by cuboidal epithelium, while the thin are lined by squamous. Third segment, the distal convoluted tubule. So this is the segment that's going to collect to the collecting tubule and then to the collecting duct. The distal convoluted tubule is lined by cuboidal cells. It does have microvilli, but not as many as those lining the proximal convoluted tubule. The distal convoluted tubule is permeable to ions and to water. Now, there's an important feature of the distal convoluted tubule, and this is going to be similar with the collecting duct. The cells lining the wall of the distal convoluted tubule are responsive to hormones acting on the process of urine formation. Now, I don't know if you remember way back in the beginning of the course, in the endocrine system chapter, we talked about some hormones, for example, ADH, antidiuretic hormone, and aldosterone. These hormones work by affecting how efficiently the kidney reabsorbs water or ions. And how they work is on these cells in the wall of the distal convoluted tubule and also the collecting duct. These hormones stimulate these cells to make more ion channels or water transporters. And because of that, these cells can reclaim more ions or more water to the body instead of letting it pass into the urine. So one of the main functions of the cells lining the wall of the distal convoluted tubule and the collecting duct is sites of action of hormones that are going to regulate absorption of some substances, ions and water, back into the body and prevent their loss in the urine. We'll talk more about that when we talk about the physiology of the nephron. All right, so a brief slide to talk about collecting tubules and collecting ducts. What's the difference between those terms? So each nephron, each renal tubule, the distal convoluted tubule is going to connect to a collecting tubule. So there's one nephron is connected to one collecting tubule. And then you have a bunch of nephrons that are hooked up to a single collecting duct. Collecting ducts, therefore, receive filtrate from numerous nephrons, whereas a collecting tubule is receiving filtrate from only a single nephron. Okay. Collecting ducts then are going to run through the cortex, then through the medulla, and they're going to end up connecting to the minor calyces in the renal pelvis. So they're kind of the highways of conducting, at this point it's called urine, to the renal pelvis at the minor calyces. Okay. This slide is just a peek at the histology. If you look at a section in the cortex on the left versus the medulla on the right, basically what you see in both of them are a lot of tubes in cross-section. So all of these are tubes. However, they're different tubes. In the cortex, you will see a lot of distal and proximal convoluted tubules. And you may also see an occasional renal corpuscle, right? So those are the main structures you'll find in the cortex. In the medulla, the tubes are going to be the loop of Henle running through the medulla, or the collecting ducts. So these really large tubes here are the collecting ducts, and the smaller diameter tubes are going to be the loop of Henle. So there you might see the difference between the larger segments and the smaller segments, thin versus thick segments of the loop of Henle. Okay. All right, I'm going to end this video here, and in the next video I will talk about two types of nephrons, starting with this slide.